according to Britannica, there are about 144,000 known fungi species known to the kingdom of fungi. This includes yeast, rust, smuts, mildews, molds, mushrooms, and other fungi-like organisms. And mushrooms, although with somewhat of a bad reputation, they are actually of great environmental and medical importance and use. Thus, over the past few weeks, we used 3 different types of fungi and tested how different forms of commonly found biomass could actually affect their growth and development.
So our question for this project was essentially, how do various natural liquid nutrients affect fungal growth compared to industrial liquid nutrients?
So our speech is going to be formatted where we're going to tell you the research that we went through before we started the experiment, what our hypothesis is, and then what we actually got from our experiment, and some roadblocks we may have encountered and what to actually conclude from what we have.
All right, now here's some background information on our project. This experiment is based off something called a liquid culture, which is a substitute to a typical solid substrate. With liquid cultures, you can get around having to buy a 3-pound block of mycelium and wait 5 days for shipping, and instead just buy a $12 syringe that already comes with mycelium, inject it into a specialized jar like this one, and now I can just take more of this mycelium from this jar anytime. I'm going to start growing more mushrooms. Now, in most industrial settings, people use a solution called a 4% light malt extract solution. This is basically a tablespoon of light malt extract powder for every 100 milliliters of water. So we used 400 milliliters of water, so 4 tablespoons of light malt extract for our control jar, and then use 400 milliliters of water run through various blended versions of natural biomass, like weeds, grass, and manure, to create these other jars.
So now we have the light malt extract, which is very highly water soluble. As a matter of fact, if I were to put some in my hands, it's so water soluble that it would start clumping in my hands. As you can see, it's already clumping and sticking. And so we used light malt extract, grass, weeds, and manure to create our different substrates. And we used 3 different species. We used the pearl oysters, the reishi, and the Pestaloteuopsis microspora.
Seen in order down there.
Yep, in the exact order as found up here. Now, the pearl oysters are mostly edible, and that's really their only function. And then you have the reishi, which have medicinal properties and work well as a styrofoam, as seen here. And now you also have the Pestaloteuopsis microspora, which is found in the Ecuadorian rainforest, which is known for eating plastic.
So out of the 3 liquid cultures, not counting the control group, our hypothesis was that the manure would affect the mushrooms the most in a positive way. According to the EPA, manure is known to contain chemicals such as nitrogen and phosphorus and other nutrients that plants need in order to grow. Compared to weeds and grass, it has many more nutrients, and we also believe that in comparison, when comparing the weeds and the grass, the weeds would provide more nutrients for the fungi, allowing more growth in the grass. According to Livestrong, this is because although regular grass provides nutrition and fiber for animals, the high cellulose content provides few nutrients for other organisms. Overall, the manure would affect it the most, the weeds having the second best results, and the grass coming in last.
Now, in terms of fungal growth, what we're actually looking for is the development of something called hyphae. Now, hyphae is essentially thick-walled tubes that contain cells of fungus, as you can see right here. Now, these tubes end with a long conical shape, which slowly elongates and extends, which is how it grows. Hyphae also occasionally branches off, as you can see here, and this essentially can create a long, tight-knit web of these tubes, which creates a larger overall organism called mycelium. Now, here's a short video showing our results over essentially a week-long period in our petri dishes. As you can see, this one spiked in growth, but then slowly starts to decrease and show contamination. This one shows structured and slow but steady growth. And the rest of them just kind of flounder, as you can see.
So the first group that we looked at was our control group. First, we looked at our Pestaloteuopsis microspora, which had heavy structure, branching visual hyphae, and experienced a large amount of growth. The Ganoderma lucidum had thick and really viscous growth, which was the one that was pointed out in the video to have the biggest effect on itself. And our pearl oyster had visible growth as well and visible hyphae, but it was not as noticeable as the other two groups. As you can see in the picture here, the Pistalotiopsis microspora experienced a lot of some contamination and does not look so hot. And in these pictures here, you can see visible branching hyphae, even though the quality is not as good as we thought it would be. In our second group, this was arguably our best result of the whole experiment. The Ganoderma lucidum had thick and really heavy growth. And lastly, our pearl oyster, as you can see, was not as noticeable in change as the other two groups, but there was still visible growth and visible hyphae when we looked at it under the microscope.
And the second liquid culture we looked at was weeds. For the Pistalotiopsis microspora, there was scattered branching and we saw signs of growth, but however, later it got damaged. And for the reishi, we saw visible structure growth and hyphae growth under the microscope, and there was a lot of branching. And for the pearl oyster mushrooms, there was actually very little growth as there was scarce damaged clumps of branching and hyphae. And so here we can see there is a bunch of growth, but it got damaged, as you can see here, and there wasn't that much growth visibly. And for the reishi, there's actually a ton of growth here, actually the best out of the weeds. There's a bunch of hyphae and branching here and germ tubes, and over here we can see a beautiful structure that developed throughout our experiment. And for the pearl oyster, this also got fairly damaged, and there's not really any hyphae or visible branching structures you could even see. And over here in the visible thing, you couldn't really see anything either.
As for the grass, the Pistalotiopsis microspora had a large flat plane growth, but that's really the best we're going to see out of the grass. The reishi had small amounts of growth, but the hyphae seemed to be disturbed and was pretty unstable. And then we also had the pearl oyster, which had small growth, but if it had more time, it might have the potential to grow. So here's the Pistalotiopsis microspora. As you can see, there's not very many hyphae here that's visible. It's not the best quality, but this is probably the worst one of the grass. And now here's the reishi. As you can see, there's not a whole lot of growth going on here. There's small pockets of growth, but there's nothing much here. And then you have the pearl oyster, which is pretty hard to tell, but it does have some good growth in the petri dish, and it does have some potential. It just needs time.
The last group that we looked at was the manure, which is, if you remember in our hypothesis, was the one that we thought would have the best effect of the growth on the growth of the fungi. When we saw these results, they were actually surprising because they were exactly the opposite of what we were expecting, and there was not a lot of changes at all in what we were looking at. For the Pistalotiopsis microspora, there were no visible germ tubes or hyphae when we looked at it under the microscope. And for the Ganoderma lucidum, it was scattered, but the hyphae was still intact. It was still visible. And for the pearl oyster, it was the same thing. The hyphae and the germ tube were almost undetectable, but they were still present. As you can see, the quality in these images are not as good as we thought they would be. But under the microscope, you can still see that there was nothing. There were no visible germ tubes or hyphae. In the Ganoderma lucidum, there was a little bit less to see compared to the other groups, but the hyphae and branching was still there and still intact. In the pearl oyster, it was, as you can see, it was almost unnoticeable, but we looked closely enough and the hyphae and the germ tubes were still present.
Alright, so overall, our comparison results basically shows that the only type of substrate that really did feasible amounts of growth was the control solution with light malt extract. This actually makes sense because light malt extract is used pretty much everywhere in an industrial setting, and there has to be an obvious reason for that, namely that it grows mycelium really, really well. However, it was really surprising how poorly the manure filtrate did because it's so rich in chemicals that we figured it would be a ripe space to grow mycelium on. Another surprising thing was how well the grass performs, because it actually had noticeable amounts of growth and preserved preserve the structure of the mycelium, even if it didn't perform very well. The weeds mostly did better in all the other areas, but the grass kept it intact, at least to a certain degree.
And our experiment, however, did not come clear of roadblocks. There are many things that showed as problems for us. One of the hugest problems we had was time. As time played a huge factor in the growth of the mycelium and fungi, as if we had more time, perhaps we would have seen different results. As Jackson said, for one of the grass liquid cultures, if it had more time to grow, maybe we would have seen more growth and hyphae and branching within it. However, we didn't have that time. And reishi actually takes 30 to 40 days to fully develop. So if we had 30 to 40 days, maybe we would have seen more growth and development in that type of mushroom. And time also played a big role in our other roadblocks as well, as we chose to do our experiment in the class, which gave us about 45 minutes to do 12 samples of fungi and liquid cultures and placing the fungi in the petri dishes and making sure everything was correct, which gave us very limited amount of time, obviously. So that kind of contributed to us having lab mishaps. And an example of this is us spilling some of the liquid culture out of some of the petri dishes, meaning that some of the samples of fungi had different amounts of liquid culture, which could have affected the growth of it or stunted the growth. And the last thing we kind of experienced was contamination. As Hadley showed in the control group, we had contamination in one of the samples, and it molded and had different types of growth, which we didn't really expect to happen, and that could have affected our results. And maybe the manure, because it didn't grow as much, it could have also been contaminated, but we don't really know.
So after we visited our hypothesis, it wasn't fully correct. We got about half of it right. We did say that the grass was going to be behind the weeds, and yes, the weeds did outperform, but the grass still did decent in some ways. And we also did say that the manure was probably going to do the best, which it did the worst, and the control did the best, so we got about half of it correct.
And to somewhat conclude, like Jackson said, our hypothesis was both incorrect and correct, considering the fact that the manure failed to support forms of any type of growth really, and the grass and weeds performed basically as much as we expected it to. And the basis of experience and from our results, what we can conclude is that this experiment basically reaffirmed the point of using light malt extract as a liquid culture or just as something to help fungi grow, because it's an industrial used, it's the main thing to use for fungi growth in the industry of mycology.
Now, the fact that the grass and weeds were actually able to support noticeable fungal growth was a pleasant surprise, as our group mostly expected not much tangible results to be yielded, but it did surprisingly well and kept the hyphae intact. Now, future experiments will likely explore the viability of using other forms of sugar and carbohydrates as opposed to light malt extract, because there's several different types. There's sucrose, fructose, glucose, and so many others that might outperform light malt extract, in fact. Now we're done with our main slides, allow me to digress for a moment and explain the importance of fungi. Fungi can be used for pretty much anything. It's a crucial part of every single ecosystem that anywhere in the world. It is very, very, very important. In fact, some companies like Ecovative are dedicated to creating different uses for fungi, for like insulation, for housing, leather for shoes, shipping and packaging, and even mycelium bacon. That is pretty crazy. Now, if you purchase these kind of products instead of traditional products, it will greatly benefit the environment and help steer away from polluting practices like heavy-duty livestock that takes a lot of land and water. Now, if you want to get into mycology for yourself, anybody can start by identifying mushrooms in their own backyard, going online to forums, or if you want somebody to talk to you, come talk to me. I've got plenty of mushrooms to share.
Thank you, and here are our references.